Both a privilege and a pleasure to stand before you today to introduce Professor Conrad. As I know Dr. Indah will introduce her later formally, but probably I'll put a personal touch to it. Although I graduated from the same university as where Pat was as a member, we did not cross paths while I was there. I know her as a faculty member, but definitely she did not know me at that time. Maybe we have bumped into each other at the UC Davis Vet School Cafe called Scrubs, or strolling at the Arboretum, the beautiful botanical garden where one of its many entrances is at the site of UC Davis Vet School. And through the effort of our ex-head of department, Associate Professor Dr. Siti Harani Bejo, I think she's not around today. Professor Conrad was appointed as our visiting professor, and she was supposed to come last year. Okay, but because of several issues, it did not materialize until this year. So when I first met her at UC affected every corner of the globe has underscored the devastating consequences of ignoring the health of animals and the environment. By adopting a one-health perspective, we can better prepare for and mitigate such crises in the future. Climate change, habitat destruction, pollution, and other environmental challenges not only affect the well-being of countless animal species, but also pose direct threats to human health. These issues demand our attention and a coordinated approach. Embracing One Health is not only a necessity, it is an opportunity. It is an opportunity for us to harness the collective power of science, policy, and collaboration to create a healthier, more sustainable world. It's an opportunity for us to make informed decisions based on the best available the health of the animals and the health of our planet. I think Pat will share with you today her life experiences working collaboratively, utilizing the One Health approach in solving the issue related to parasites. So I don't want to take up much of her time. Knowing Pat, I know she can easily inspire people with her stories. So let us hear from her. Thank you.
Thank you very much, Dr. Farina. So now is the moment that we have all been waiting for. Before I call upon our guest of honor, the star of the show, allow me to briefly introduce her. So she's charismatic, confident, and fierce. From the first time I met her, I know that she is not the woman to mess with. There is never a dull moment when you converse with her. So she is a distinguished professor of parasitology emerita at the Department of Pathology, Microbiology, and Immunology at the School of Veterinary Medicine, University of California, Davis. Professor Dr. Patricia Conrad, beginning July 2021 after her retirement until present. Her specialty is on protozoology, One Health, and global health. Previously, she held many top positions, of which includes the former co-director for the Global Health Institute and as the former associate dean for Global Program. She graduated from the Colorado State University with a degree in Doctor of Veterinary Medicine with high distinction honors, and later received the Marshall Aid Commemoration Commission Scholarship to pursue her PhD in protozoology and tropical animal health from the University of Edinburgh, which is also my alma mater. Hello. So this, among many others, and awards, long list of awards, lectureships, scholarships, fellowships. Note that all are in plural, received throughout the years, including the latest one last year, a grand one. I will not spill the beans, so I will let her share her amazing achievements in her own words shortly. She has a very, very long list of significant research discoveries, spanning from discovery and characterization of new hemiprotozoal species affecting humans to other protozoan species in various animal hosts with important public and One Health impact. Her peer-reviewed publication record is extensive, more than 230 journal articles to be exact. She has a remarkable list of countries in global work experience, jet-setting between different locations, cross continents, from the UK to Africa to Asia and back to the States. So I will not be spending more time. I do not want to spoil the party. So I will give the honor to the speaker to tell her One Health journey in her own words. Ladies and gentlemen, let's put our hands together for distinguished Professor Emeritus, Dr. Patricia Conrad. Okay.
Whoa, thank you so much for that wonderful introduction, Dr. Inda, and also Dr. Farina. Thank you so much. I actually wanted to start this out with expressing my gratitude to all of the faculty, staff, students, veterinary students, and postgraduate students here at UPM who have made me feel so welcome. This has been a wonderful visit. I've learned so much. And as a sign of my gratitude, I would like to share this very personal journey with you. Are we good?
Yeah, you can visit.
Oh, okay. I thought maybe I needed a translator here. Sorry, my Malay is not very good. And thanks so much for coming. For both those of you who are here in the room, which I love, contact with real people, but also those who are joining from a distance. So we need to go back to the slides. You hit a button there. This is where a little bit too much technology, perhaps, for me. We have capable people here. Okay, do we have to have that picture on here?
Yeah, because we have live stream.
Okay.
They want to see your face.
Okay, that's good. That might cover up a few things, but we'll just do the best we can here. So a very personal journey is what I would like to take you on. And it's one that even now, as I've retired, I look back and I am just amazed that it turned out so well with so many wonderful people and the discoveries that we were able to make. And as you just heard, I would attribute a lot of that to this One Health approach. Even before it was called One Health, this collaborative approach to research and getting the engagement of people. I have been blessed by having incredible colleagues and students, both male and female. But as you'll see, I've been really excited about how young women have been able to advance in their careers. So let's see, we can advance this. There, hold on. All right. So One Health, when people think about One Health, and probably not so much all of you that are related to the vet school, but out in the big world, I think they most often think of zoonotic disease, right? Because that's really what drew recognition to the concept of One Health. The idea that you cannot look at human disease, especially infectious disease, in complete isolation without thinking about the environment that people share with animals and the impact we have on that environment and that it has on our health. So these are just some of the zoonotic, transmissible between animals and humans diseases that people think about when they think about One Health. But I hope, if nothing else, when you leave, you'll not only know a little bit more about me, and that would be fun, but also about this concept of One Health. And I like this diagram that we put together at UC Davis. You can think of it as a big chunk of stilled cheese or cake, if you love sweets, as I do, and the many layers. And this component of looking at the interconnection between animals, humans, and the environment is right here. But by taking that approach, you can apply this approach to so many different problems and challenges: nutrition, food production, disaster, the way you approach disasters. We're doing that with the wildfires in California, healthy water and air, zoonotic disease, and also, of course, parasite challenges and viral challenges. And I always stop when I think about One Health. I think about the veterinarians and the veterinary students and the scientists. Maybe you don't have a veterinary degree, but are engaged in looking at veterinary problems, because that's the environment in which I live. And I have to ask myself the question, in some ways, are we not natural One Health practitioners? Because coming into this field, we look at all these different animals, their owners, our clients, the people, and the environment. So it may just be, as I reflect, that we are, of all people, probably those that most naturally understand One Health. This may, in fact, even be our superpower in our profession, is that we have that understanding. And as you know, with any kind of superheroes, they all have an origin story, right? I saw a lot of support for Spider-Man in this country, right? And when you think,
I'm sorry to interrupt.
no problem. Reflect on your favorite superheroes while we're doing this. Is Spider-Man popular here?
Yeah.
Yeah, that's what I thought. And we all remember how Spider-Man got his powers, right? From the bite of a spider. That's his origin story. Okay, we think we're set? Okay,
Sorry.
thank goodness for a technologist.
Lemak dia dekat ujung. Mana matanya? Ya tu. Ni dah pula ya.
Stick with us, folks.
Ni ya?
Ya.
Okay. Ni slide mana?
I love that emoji.
Okay, tak pernah kerja.
Lemak kecil.
Sebut evaluation.
Okay dah.
Okay.
That's okay. I can advance. We good? Good? All right.
Okay.
So some pretty powerful people here. I hope we're all feeling powerful, right? With our superpowers. So origin stories, because, and I bring this up because basically, I think stories are very compelling, and I think it's good. We all think we don't have an interesting story, but we all do. So think about what attracted you to veterinary medicine and veterinary science, or maybe some of you are in human medicine or environment. What attracted you to that in the first place? All right. So now my story. Well, I never expected to be here. I don't mean just in Malaysia. I mean, as a research scientist, as an academic. And as a little background, I grew up in Colorado. All right. That's Pikes Peak, which is the Colorado Springs. My hometown is at the foot of Pikes Peak, the foot of the Garden of the Gods, these beautiful red stones, if you ever get a chance to go there. And I think these pictures reveal from the time I was a very little girl, I wanted to be.
Dia punya ni dah banyak dah hari. Sekejap. Gua share.
We thought we had this all worked out before we started. Sorry.
Mana ni? Ya, dia punya banyak lari ni.
Hopefully our people online are sticking with us through this. Be a pity if you all got up and left while we're in the midst of sorting this out. You might be able to, I'll pick out me. All right, there's me. All right, that's my big brother. This is me right there. And that was even when we lived in town, and we didn't. Okay,
All
and now we have music.
Sure.
Oh. Okay, so here we are, taking you back a minute, okay, to Colorado Springs in Colorado, in sort of middle America. We like to think of ourselves as the West. And I dreamed of being out in the country, being a cowgirl. I don't know if that's much of a concept, but it was because of my love of horses. And this was not my horse. This is just a little horse that they brought by so you could sit on it and have a picture taken. But as our family grew, and we had 7 children, my mother had 7 children in 10 years. So my father decided we should move out to a bigger house out in the country. And that, of course, encouraged me even more, because with a bigger property, we could have more animals. So I essentially grew up with a menagerie of animals. I mean, up to 20, 25 cats at a time. We gave away our kittens, but we had a lot of cats, goats, baby goats that we used to let run around the house, chickens, a lot of horses. This is me right here with my sister, good friend, and one of my brothers. And so this was, my dad he had 5 sons and 2 daughters. He thought, what is a sport? What is an activity? What is a sport? Is that back? All right. What is a sport? What is an activity? I can step back a little, that we could all do together. And he came up with the idea rodeo. How many people have heard of rodeo? All right. So that's really a game for cowboys. It includes, for men, riding broncs and steers and horses. And for women, fortunately, we don't have to do that. For me, it was roping calves and tying goats. So this was a family activity, and you can see all of us there. My sister was the queen. There she was. I was the support team. Right there. All right, my sister and myself. And these are some of the horses we had. But I loved it so much, and it was a great sport. And that's what they gave. If you got the top honor, you got a saddle. So you can imagine, with that kind of background, I loved animals, a wide variety of animals. And my dream was, I think from the time I was 8, to be a veterinarian, and I could take care of all these different animals, not just dogs and cats, but a wide variety of animals. And so I managed to go. I started out at a community college on a rodeo scholarship. And then I went to Colorado State University, where they had the veterinary school. And I was fortunate to get accepted there. And I loved vet school. Knowing I wanted to be a veterinarian, everything was exciting. Everything was something I wanted to absorb. But I distinctly remember there was one lecturer who came in and talked about this disease, East Coast fever. How many people have heard of this? Right? It's a disease caused by a single cell parasite that's related to the one that you have here in Malaysia, Theileria. You have Theileria orientalis. Well, this is a much more pathogenic parasite that actually affects cattle and ends up with infected, fully susceptible cattle having mortalities of over 97, 98%. So a really important disease of cattle. But as this professor who had been in Kenya gave this talk, all I could think of as a vet student, it was the only time I did, I thought, why? Why is this person taking time in our busy curriculum to talk about this parasite that I will never see, a disease that I will never see? And I mentioned that to vet students because we all have something like that. We think we'll never see. All right, so just wait till you see what happens. So I was moving on through vet school. And we had an opportunity when we were in vet school to work through the summer, our senior year, and take our break afterwards if we wanted to. And I did for large animal. So I took in that break right before the final phase of my senior year, I took what I had never taken before, and that was a vacation. And I am now a big advocate of vacations. You know, Americans were not very good at vacations, but I am a big believer because it was during that vacation that I went with my older brother to Wyoming, to the Grand Tetons Mountains, and it was taking a little pause there after I had a conversation with my brother. I had planned on going into practice, making money, going back and supporting my family because my father had lost his job and we had a lot of children still there. And my brother said, no, you don't have to do that, Pat, because I'm happy. I will continue doing that. You should do what you want. And that question, what do you want, was one that I hadn't thought about since I was 8 years old. And it was because of that and my brother and taking that time to pause that I had my aha moment. And that aha moment was very strange, unexpected for me, because the very first thing that came to me was I need to go and live in another country, not travel. My family didn't have money for that, and I was putting myself through vet school, but to travel and live in another country, work in another country. How in the world was I going to do that? I didn't know anything about other countries. Well, the thing that came to my mind was Africa, because we talked a little bit about Africa. Colorado State University had been working in Kenya, and it occurred to me, I really liked cattle, sheep, and goats. And in a number of different places in different parts of the continent of Africa, these animals, cattle, sheep, goats, are critically important. And that's something that veterinarians know about. I knew about, and I loved them. I didn't know all their diseases, but I knew something of cattle, sheep, and goats. And I knew that if you could improve the production of livestock, of food animals, that you could actually improve the nutrition and the health of humans. Now, how was I going to do that? How was I going to get there with no money? Well, I started looking for ways, and fortunately, I saw a little ad up on the bulletin board outside the dean's office. Sometimes this is the way it happens, serendipity. And it was about these Marshall scholarships. They're like Rhodes scholarships, but you can be a little bit older and you can study anywhere you want to in Great Britain, in the United Kingdom. So this is what I chose, the Center for Tropical Veterinary Medicine. Just found it in a book, all right? And it sounded right. I figured they speak English here, all right, because I didn't speak anything else. I'm not linguistically talented. And they're working on problems that occur in tropical areas where livestock are really important. So this is what the Center for Tropical Veterinary Medicine at the University of Edinburgh looks like. It's still there. I went and saw it just last year. It's kind of running down, but it's still there. And this is where sitting in a classroom, not a little smaller than this one, I heard a lecture about East Coast fever. Remember that disease that I mentioned? But this time it was by somebody who had spent his entire life working on East Coast fever, trying to develop a vaccine to prevent this disease in cattle. Duncan Brown was his name. And this, I just captured his passion and talking about this parasite that was transmitted by ticks. You can see there up the top, there's all these ticks that are on the ear of cattle, Rhiphocephalus appendiculatus, and they inject the parasite. And ultimately what kills the animal is pulmonary edema. But it's a fascinating parasite because when the tick feeds on the animal, it injects a little stage of the parasite that goes into lymphocytes, and it transforms those lymphocytes, immortalizes them in some ways like a cancer virus, but an oncogenic virus, but by a different mechanism, but causes the lymphocyte to divide under the control of the parasite and to keep on dividing, so that if you actually took an infected cell from a wild animal or cattle that had the parasite, it would keep on dividing in culture, immortalized. And this was just, to me, absolutely amazing that any parasite could take control like that. And this began a very passionate love affair for me with a parasite. So this is Theileria up here in a lymphocyte. That's the Theileria parasite right there, multinucleated. And I was passionately intrigued by this parasite. So after I did my PhD in Edinburgh, and I did that in less than 3 years because that's all the scholarship was. I got a postdoc here at the International Laboratory for Research on Animal Diseases in Nairobi, Kenya. It's now called ILRI, but it's still there, and it's an amazing research lab. So I went there because this was a laboratory that worked on two things: trypanosomiasis and this disease, East Coast fever. And the whole focus in terms of the East Coast fever was to try to develop a vaccine that could be given to cattle to prevent them from getting the disease, because that would have a big positive economic impact on the eastern part of Africa, where the disease is so important. Now, a disease had been worked on by my major professor, actually, an infection and treatment method that worked very, very well on cattle if they were immunized and kept in captivity. But cattle in Africa aren't kept always in captivity. They graze. And if they grazed in areas where there were wildlife, like this African Cape buffalo, bit different from your Asian buffalo, but they are carriers for Theileria. And if ticks feed on them and then are transmitted in the next stage to cattle, even vaccinated cattle succumb to the disease of East Coast fever. So I was fascinated with the disease. I had come with knowledge from my graduate work. I joined a lab where there were 2 or 3 labs that were working on this. And it was an amazing group of international postdocs and scientists. And this is where I learned my first very important lesson. Back then, we didn't call things One Health, and people weren't collaborating all that much. But here was a great opportunity to do that. And what I learned was that when you take an important problem or challenging situation, a complex challenge, that these two things can be powerful motivators for collaboration. And that's exactly what happened. I was put in an environment where I was the parasitologist, but there were immunologists, tissue culturists, cell biologists, epidemiologists, molecular biologists, people of all different backgrounds and expertise, mostly postdocs like myself, that had great ideas, and they had skills that I didn't have. And we worked together, and by having these different perspectives, not just the disciplinary perspectives, but working in an international environment where we had people coming from different cultures, from different backgrounds, that dynamic of different ideas really had an impact. And for me, I had a 4-year postdoc there, during which time anyway, I also had a personal life. I didn't just work all the time, and yet it turned out to be incredibly productive. And I put this in here because this is what happens when you are working with a team of people, just sort of spontaneously come together. And these were all the different things, areas that I was able to work on that I would never have been able to do by myself. I didn't have molecular techniques or able to characterize genes or look at drugs or any of that. And it also turned out to be very productive in terms of getting this information, what we discovered, out into the literature with publications. So lessons learned from my postdoc, the big ones, is that collaboration does not require a leader. You do not need the head of the lab or someone telling you this is what you need to do. You need to work together. We can come together and bring the ideas and bring the expertise, and that mutual trust and respect are essential for successful collaborations, not just skills, but mutual trust and respect. And also, collaborative research is fun. You can really have a great time doing it. So from that experience, when I came to ILRAD, I didn't think I'd ever be a research scientist. I didn't have that much fun in my PhD. I was going back to be a practicing veterinarian, but this changed my whole understanding of what research could be. Research was, for me, it was transformational in more ways than one. My 4 years in Kenya, not only my attitude towards research, but I also got married and I had my first son there. Here he is, Alex Markham, and dancing. That's how, if he made any noise, that's what we do. We start dancing. So it was a transformational experience. I loved it. I was offered a scientist position to stay there, but there comes a time when you just know you got to make a move, or you maybe don't have to, but you want to make a move, not because you're unhappy, because you could be happy, but because there's something missing. And for me, my out of Africa move to California was really motivated by my interest in working with students. So that's when in 1988, I got the faculty position at the University of California, Davis, was to be able to work with students because I love it. Now, the challenging side, the scary side of all this, and I mentioned, because we all have scary sides, was that I had never written a grant. I had no grant writing skills. I had never taught a class, all right? And academics have to teach, right? I never had taught, and I had no plan for my research. I knew that I was not going to be able to work on East Coast fever because it didn't occur in the United States, and they didn't even want you to bring slides into the country. So I had to find something else. Fortunately, they didn't ask me what I was going to work on. So we don't do that anymore. We do ask people, but they didn't ask. And so I got the job. And when I pitched up there, I had to figure out, what am I going to do? So I knew they had one thing in California that I absolutely loved, and that's cattle. I love cattle. And there's a lot of advertising about the happy cows of California. And there are some happy cows in California, some not so happy, but there's some happy ones. And within the first few months that I was there, I was confronted with a problem. And that problem came to me by these two pathologists, Brad Barr and Mark Anderson, who'd been working in the state diagnostic laboratory system. We have a great one in California, and the main lab happens to be located on the same campus with the veterinary school. So that cross-pollination was fantastic. They came to me and they told me they were having a problem that they had over the last 6 months prior to my arrival. These mid-gestation abortions. California is a big dairy state, often on top 1 or 2 dairy states in the United States. And so this was big because if dairy cattle abort, they can't produce milk until they get bred again and they actually have a calf, right? That's how it works. So having mid-gestation abortions had a big economic impact. And they, as pathologists, had been looking at those fetuses, and what they were seeing was this diffuse, non-suppurative inflammatory set of lesions. I don't know if you've... okay, somebody needs to mute themselves, Sarah. So somebody on our outside audience.
Saya nak suruh orang buat.
Okay. All right. Anyway, do we have any pathologists in the audience? All right. I don't know that, but I got to be a little bit of a pathologist. We all get pathology when we're in vet school, and these non-suppurative lesions like that were the first clue, because that's characteristic of viruses and protozoal parasites. And then they also, even before I got there, they had seen some of these. Do you see right here, this small structure? This looked like the cyst, the tissue cyst of Toxoplasma gondii, if you've heard of that, probably the most successful parasite in the world that affects all warm-blooded animals, including humans. And so they recognized that looked like Toxoplasma, but it had a thick wall, and Toxoplasma has only ever been reported as having a thin wall, and Toxoplasma had not been reported as a problem in cattle. So it just didn't fit. But I had arrived. They said, hey, you're a parasitologist, you're a protozoologist. What do you think? And that's where we started to work together. And we were able to start looking at these fetuses, at the encephalitis and the myocarditis that was associated with these protozoal infections. And at that time, about that time, Neospora was discovered in dogs. So we were able to get anti-serum and show that those abortions, that parasite that was associated with those abortions, was actually reacting to this antiserum for Neospora. And the same was true in calves, because calves sometimes were born looking perfectly normal, infected calves, but we'd also get calves that had severe neurologic problems. We necropsy them and find that they were infected with Neospora as well. And then working with epidemiologists, we went beyond pathologists, epidemiologists, we were able to show that, in fact, this Neospora-like protozoan was causing the majority of these unrecognized abortions in California, and was a major cause of abortion in California. That went on to be the case in a number of different countries. So it became recognized as a big problem. It had been there before. Sometimes it's a matter of discovering, recognizing what's there causing the problem. And that was the case. Probably our biggest contribution here was also we were the first to isolate the parasite from aborted fetuses and from calves and grow it in culture. Because once you have that, then you can actually fulfill Koch's postulates, as you may have heard. So you can see a parasite or an infectious agent associated with the disease, but can you reproduce that disease with that parasite or that infectious agent? And we were able to do that. We're also able to develop serologic tests that we could use. So it turned into a big team effort. We were able to contribute a lot to the whole Neospora problem investigation, and even working towards a vaccine, which was not totally efficacious. No vaccine has been efficacious against Neospora because it's an intracellular parasite. It's very, very clever parasite like Toxoplasma. So up to this point, we were talking about One Health. So I had been working on East Coast fever and bovine Neospora, and One Health, that wording had not been invented, but in reflection going back, would you say that was One Health? Was that a One Health approach? Well, I would say yes, it was, because we were looking at food producing animals that their health really affects the health of human beings. We were very collaborative. We were very collaborative and very multidisciplinary. If you look at the group of people that ended up working on Neospora. Zoonotic potential, this is a little known fact that actually kind of has gotten buried in the literature. People don't want to face it, but we did actually show that Neospora isolated from infected cattle could, when used to infect rhesus macaque monkeys, caused congenital defects and death, just like Toxoplasma. So human infections of Neospora have not been identified, but I say keep the question open. All right. You don't know. And if you don't think about the possibility, you won't look and you won't discover. So we were happy taking off with Neospora, but another exciting thing happened that I think you can relate to even better. And that was within the first 2 months that I was at Davis, a clinical diagnostic laboratory sent me a blood smear that had been made from a dog in Southern California. They had been having a problem with anemia in dogs. They thought it was autoimmune hemolytic anemia, but then with anemia, thrombocytopenia, fever, and splenomegaly. Is this sounding a little familiar? For some of you that look at your dogs. Then this clinical lab sent me this slide. And if you look carefully on this slide, it's kind of hard because it's kind of bright lights here, but in the red blood cells, you can see little tiny parasites. Now, when I saw that, my heart jumped for joy. I thought, oh, Theileria, maybe they have Theileria. Well, there isn't a Theileria in dogs that we know of. So what does look like that, I had never seen it before, are Babesia parasites, some Babesia parasites, specifically the small Babesia. Now, at that time when we were doing this, Babesia canis was the large Babesia. Now we know that there's Vogelii and some of the other large Babesia species have been recognized, and you can see them. They're much easier to identify right there. Oops, sorry, in that slide. Oops, the little pointer's a little hard, but the small Babesia, at that time, there was only one small Babesia in dogs that we knew of, and that was Babesia gibsoni. And so we called this parasite Babesia gibsoni. My very first PhD student, who actually had come from Japan, he got on this right away. He made some really significant contributions. We're working as a team here, but in terms of developing the diagnostic tests that were necessary, we started doing the things that veterinarians do, and that is trying to figure out how to stop it, how to treat it. But we also stopped to ponder. And I think this is an important question always to ask. What about zoonotic potential? All right. Is there any chance that this parasite could be infecting humans? And at that time in the United States, there was only one Babesia parasite that was known to have zoonotic potential, and that was Babesia microti. You can see the life cycle here. This is a parasite that normally survives in deer mice and small rodents, and is transmitted by ticks, and only occasionally can be if one of these ticks that's picked up the parasite from a rodent and has molted to the nymphal stage, particularly feeds on humans, they can infect humans. So there weren't a lot of cases at that time. There's since it's become much more recognized disease, but that was the Babesia that we knew about. So that most of those cases were in the Midwest and the eastern part of the United States. But then our big chance came in 1991, where there was a case of Babesia in a person in Washington, D.C., a 41-year-old man that actually had severe hemolytic anemia, and they sent the slides to the CDC. Originally, the disease was misdiagnosed as malaria because the parasites look so similar, but then they started to think about it. This person had no history of being out of the United States and had not been in a malarial area. And they did see this little small parasite, but he didn't have any antibodies to Babesia microti. So this is where they thought, what about this crazy idea that Pat Conrad at the University of California, Davis has been talking about, that maybe there is another small Babesia that might be zoonotic. Maybe it's even this dog Babesia, as we pondered. So we did that work, and we showed that, in fact, and we won't go into details here, but molecularly characterizing the WA1, that's what we called the isolate because we didn't know what to call it. WA1 was actually more closely related to Babesia gibsoni, which you have in this country, okay? But this was the one that we had from dogs, Babesia gibsoni, we were calling it, and more separate phylogenetically, molecularly from Babesia microti. So we knew that it wasn't the same. We also knew that it was closely related to the canine isolate, but did not infect dogs. So there were a few other cases after that, but we got very poor samples from them. The clinicians oftentimes just didn't know where to send them. And so it wasn't until 1998 that we had another big breakthrough. We were actually at a tick meeting with all the tick people that did research and worked for ticks in the public health department. When we got the call, everybody's phones went off at the same time. It was very exciting, like the stuff from movies, that ding, ding, ding. And they answered. And it was because of this newborn baby at a hospital in Oakland who had been prematurely born. And oftentimes those babies require blood transfusions. So they actually had given 2 blood transfusions to this little baby and noticed the baby was developing anemia and made blood smears, fortunately, and stained those smears. And look at that. Aren't those the most beautiful parasites? If you like parasites, and I do, there they are right there. And this is your classic Maltese cross form, all right, that you've seen on Theileria, because these small Babesia are very closely related to Theileria. So right there, dividing into 4. So very exciting. When we typed that, we found out that was very closely related to the WA1 parasite that we had found in that person, that 41-year-old man in Washington State. And so with that, and we took 10 years because we had to get all the information, and we went around and discussed this with a lot of parasitologists. I wanted to be very conscientious, but we did get everybody's okay that this was a distinctly new species. So we named a new human Babesia species, and we named it, I named it after my former professor, Duncan Brown, Babesia duncani. And there he is. He's in his 80s now. He's quite a character. And if anybody's been to the CTVM at the University of Edinburgh, you may have met him, but an amazing person. And so this was a way of honoring him. So what lessons have I learned from this? 1, challenge the dogma. Okay. If people say, and they were saying back then, that Babesia were very host specific, that they only stayed in their own host, we knew that wasn't quite right, because Babesia microti was transmitted from rodents. All right. But if somebody tells you, oh, it's this way, especially in protozoology, but I think in anything, question it. Are we sure? What if this could be a new parasite? What if this could be transmitted in a way that we hadn't imagined before? So for me, that was important to do. But out of all of this, there was another really important lesson that I think all scientists, we need to be aware is perfectly all right. There's nothing wrong with being wrong. All right. And my big example of that, I mean, I've been wrong on a few things, but this, my sort of really got it to you wrong, was brought to me about this dog Babesia. As we went on working on Babesia, this small Babesia of dogs that we were calling Babesia gibsoni, because that was the only small Babesia of dogs. So we called it Babesia gibsoni, was in fact the only one that had been talked about much in the United States at that time. But over the following 10 years or so, more isolates of small Babesia were found. More people were doing molecular characterization, including ourselves. And we discovered that even in the United States, there were at least 3 different genotypes of Babesia in dogs in the United States. And that, in fact, the one that we'd been calling Babesia gibsoni, when you molecularly compared it to Babesia gibsoni that you have and Japan has, and the countries where it was first described, it was molecularly distinct, and that enough so that we had to give it another name. And so one of my former graduate students, who now had gone on and was a scientist in public health, she and her colleagues, many of them who'd worked with me, decided to show Pat, you got it wrong. It's not Babesia gibsoni. So to do that, and so I'd remember it, they named it after me. So if you heard of Babesia conradii, which looks a lot and acts a lot like Babesia gibsoni, but is molecularly different, that has been named after me to remind me that I get it wrong sometimes, which was actually quite nice. This also brings me to another really important lesson from my career, and that is graduate students, here we call them postgraduates, that's what I was in Edinburgh, that they are gems, they are treasures, and they need to be encouraged and supported. And that will pay in big benefits for you and for our profession and for science. And because they really are the future One Health practitioners. They understand that we have to think about humans animals and environment all together. And Ann Kimtrup is the one who did that named the parasite after me, and is now in our California Department of Public Health, was a very good example of that. And she's gone on. That's another nice thing. I throw this in just to remind that the people we mentor then go on to mentor others. And I've already met a lot of examples. Some of you here have been the mentors of other people who are in leadership and exciting positions now. And that's incredibly rewarding. And she's doing that for people in other places, like Andrea Sway, who was able to identify go on and look at the vectors and the other hosts for these parasites with the support of Anne. So I'm going to give you a few more examples that sort of tell the story of our research and have really been led by these graduate students. I mentored them. They were mine. They're all faculty in one institution or another now. But let me just give you some examples. Antoinette Marsh, she came to me. She was a staff person. She said she didn't know if she wanted to do a PhD or not. And if she did, she'd probably do it in immunology. And I said, that's okay. If you want to just work in my lab, I need somebody to help. And in the course of it, she did get really interested. I said to her, if you ever want to do a PhD, let me know. All right. And she got very interested in a disease. You don't have it here, but we have equine protozoal myeloencephalitis. She did start dating and went on to marry an equine resident. That might have had something to do with this interest. I don't know. But anyway, she got very interested in this disease, a disease that at that time that she got started was known to be caused only by one parasite, Sarcocystis neurona. It had been identified by J.P. Dubey that this was a disease problem, a syndrome that was only caused by that parasite. And lo and behold, by looking very carefully and working with pathologists, Antoinette discovered, wait, not all of these parasites in the brains of the horses and spinal cord of the horses with this disease are Sarcocystis neurona. They don't look morphologically like that. And then when she went on anagenically and molecularly to characterize them, she found, wow, no, they actually are more like Neospora. And as a result of this, she discovered a new species of Neospora. Neospora husei, it's been named, and she named it after the man who was the director of the Center for Equine Health at our university. And we went on to develop diagnostic tests. This is one that we make available. It happens to be simple, indirect fluorescent antibody test, but very popular for the diagnosis of this disease, which is a big worry for the equine industry. All right, then the last big story that I want to tell you about took up about the last 20 some years of my career and was really, really fun. All right. Last night, I was walking out and I saw your otters in the lake, okay, by my hotel. Cute. I love otters. And I was thrilled to see this. But we have otters, we have river otters, but this is a sea otter, and these sea otters are very, very precious, precious to all the people in the states where they occur. They only occur along the West Coast, from California on up through Washington to Alaska, and they are cute, right? They're very, very iconic, and I had not been working on wildlife, except as vectors, reservoirs for parasites. But I had one of my vet students actually said to me, they're having some problems with sea otters. And there's going to be a talk here at the Marine Mammal Center about looking at the first two otters that we've been allowed to necropsy in California. Come along, Pat. I think you should start getting into wildlife. Okay. So listen to those young people. They've got some good ideas. You should get involved in wildlife. And so I went along and to try to see what they had discovered about what was killing sea otters off the coast of California. And why sea otters? Why are they so important? Well, ecologically, they're very important. In California, we have a beautiful, beautiful coast, and we have kelp, these gigantic plants that grow up and play a very important role in terms of protecting our coastal environment, protecting from erosion. They also serve as a habitat for a wide variety of different creatures, mammals, fish, all kinds of animals. And one of the killers of kelp forests are sea urchins, because they're sort of like the cows of kelp. They just really will graze until they destroy the kelp entirely. But good news is sea otters prey on these urchins. And with sea otters present, it's going to decrease the chance of them being destroyed. So that important role, the fact that they are so cute, they have become an icon for the coast of California and all of those states, Washington, Oregon. They are widely publicized. And they have had problems. So they are federally listed threatened species. Their numbers are protected by the federal government as well. And when we got involved, they were having a real problem. They do still have some problems, but with otters that had neurologic disease like this was taken an otter that was actually rescued and brought into the Marine Mammal Center. And oftentimes what they found were carcasses of sea otters. And from all of that, looking at the brain, the spinal cord, what was discovered by this was the graduate student. She started out as a vet student, then she ended up doing, we're looking at this as a project, and ended up being a pathologist and a PhD, works for the Fish and Wildlife Department. And she was one of the people that helped us to realize that toxoplasma was one of the main killers. There's that Sarcocystis pneumoniae that I mentioned in horses also kills marine mammals and sea otters, but toxoplasma was really the big killer at the time that we got involved. And I don't know how many people know about toxoplasma. I'm going to do a little quick review just in case. All right. This is a fascinating parasite. It occurs all over the world, and it has three major stages. This tiny little stage that's inside any of the animals that it affects, that causes a lot of pathology. Then when the parasite recognizes that it's going to be destroyed by the immune system starts to act. It insists, it hides in the tissues. That's the cyst stage. And then when animals, any warm-blooded animal, including birds, happens to eat one of those cyst stages in undercooked meat, they can get infected. And the third stage, this tough oocyst stage that's shown here, that shed in the feces, though, only occurs with felids, only cats. So all animals, including humans, can get infected, but only cats shed this oocyst stage, which is so tough in their feces. It's tiny. So cats, there's a special link between cats and this oocyst stage. And so here's a land animal. We're working on marine mammals. So how's this happening? How is this stage of the parasite getting to the sea otters? And it must be this stage of the parasite because sea otters don't eat any of the other warm-blooded animals that they would get infected with. So we knew that had to be it. And we also knew that if sea otters were getting infected with toxoplasma, sea otters are a marine mammal species that live very close to the coast, within about 5 kilometers. That's the same area where some other marine mammals are, but also where humans hang out, where we like to be in that water. That's also where some of the sea life, including filter feeding bivalves, like you can see here, oysters, clams, mussels, all of those, they're filtering water. And if there's oocysts in that water, they are actually going to be able to pick that up. And just quickly, if you haven't heard, toxoplasma can be a really big problem in humans. Most people get infected, only have a flu-like illness, or don't even know that they're infected. But women who get infected while they're pregnant can have very serious impacts on their babies, including from the fetal loss onto severe congenital problems like this hydrocephalus. And the other group of people that suffer are anyone who gets immunosuppressed, severely immunosuppressed. That can be HIV, it can be transfusions where you have to be immunosuppressed. And that's the problem. So we were able to identify a couple of areas along the coast of California, these big stars, where in Monterey and Morro Bay. Here's California, the state right there. This is the area where the sea otters are in California, southern sea otters. And then here were the two hotspots. What we did then was we put together a great multidisciplinary team. Look at all those different backgrounds. These were some of the people that worked with us over the last 20 years. The National Science Foundation was one of our big donors, but a lot of private donors also and foundations contributed. And then we had amazing graduate students. And I just want to highlight 3 of these because you might get a chance to work with them. This is a little while ago. They all have, their babies have all grown up. Okay. So then just to show women can be great scientists and still having babies. All right. So you don't have to have a baby to be a great scientist, but you can if you want to. All right. And so let me tell you about them. Heather Fritz, she's now the head of diagnostic microbiology at the State Diagnostic Lab and a faculty member of ours. And what she focused on was on the transcriptome and the proteome of Toxoplasma with John Boothroyd at Stanford. Really some very wonderful molecular studies that she did. But then we also needed to get out in the field. So I told you that felids, all right, not just the live domestic cats, but also our wild felids, in this case, bobcats, mountain lions, all of those can shed this oocyst stage in their feces and essentially load up the environment. You have no idea how much cats poop in the environment. It's a lot. All right. So they're pooping out these parasites, and then they're getting transported to water where, of course, as I said, humans and sea otters might be infected. And that was one of the things that Liz Van Wormer, who's now faculty at the University of Nebraska in Lincoln, she worked on as a graduate student, looking at a lot of what cats were shedding, also wild felids were shedding, and that flow of water from the land where they're defecating into fresh water and also into the oceans. We also did some nice studies on wetlands too. And she teamed up with Karen Shapiro. They were all graduate students at the same time. And Karen Shapiro, she took it a little further. She was really interested in water, and she looked at these oocysts and how they behaved in water, and what their physical characteristics were, but not just in fresh water, but also what happens in the ocean. All right. What we did a lot of work on bivalves collecting these oocysts, but she went beyond that to some really intriguing mechanisms that really broke open the whole ocean situation to us. And let me just give you one quick example. You got to ask, all right, so there are a lot of cats, they're pooping, but the ocean is huge, right? I mean, we've used this concept that, if you just use dilution of pollution, that's why we flush so much down into rivers and oceans and all. So you'd think, wow, how can all of that dilution, how do you get enough oocysts that you have this big problem in sea otters? And what she discovered was, working with other people who'd worked on ocean snow, especially marine snow, these particles that aggregate material in the ocean, that if these oocysts, as they wash into the ocean, they actually get bound up with those aggregates. And she's doing a lot of work now on plastics, and not just Toxoplasma, viruses, bacteria, how they aggregate in the ocean. And when they do that, then those are easily associated with material like kelp, and that on that kelp, there are snails feeding, and it's snails that pick them up. We thought originally, we had the hypothesis that it was mussels and clams and all of that, and they can get infected because they concentrate oocysts. But we actually, our big epidemiologic studies showed that it was sea otters that ate snails that were at the greatest risk of having Toxoplasma infections. And suddenly it started to make sense that these snails that feed all along on the kelp are picking up the oocysts, and then the sea otters eat the snails, and that's how they're getting infected. So another really big point. All right. Those were just some highlights. Support those young people. All right. Another big point that has helped us and helps keep us as scientists, and especially young people, motivated to do research, is to ask this question: how will this research make a difference? All right. Because we all want to feel like what we're doing with our lives is going to make a difference. And so I just want to give you one quick example of how the research that we did got noticed by our by one of our state legislators, when he took his young daughters to the Monterey Bay Aquarium, where we had an exhibit, and they got very concerned, and they said, Daddy, you need to do something about this. It's killing sea otters anyway. So kids also are valuable. And this led to eventually to a law that included a number of aspects, including controls on how we dispose of our cat litter. If you are ever in California, look at the cat litter and you'll see those notices. And a tax checkoff box so that on income taxes, people can give money to sea otter research. And a number of safeguards, including some laws that said that that the sea otters, that we need to protect and prevent the flow of infectious materials and toxic materials into the water that might affect not only fish and marine mammals, but humans as well. Okay, so let's wrap up here, this One Health approach. I hope I've given you some good examples. You have people working in this area, and so I think you have some good examples of your own. But when I think of One Health, think locally and globally, because it's everywhere. Whether you're coming at the problems from the perspective of humans, as people in the medical profession oftentimes do, animals, or plants. I mean, a lot of people in agriculture are very interested and are amongst our collaborators in this One Health approach. Always remember that we share environments, all right, with these plants and people and other animals, and that by taking a collaborative approach, we can really have greater impact. This One Health approach, there's no set formula. It just means working together with a broader perspective. And it can be very productive for academics and people, but also in terms of training this new generation. And I want to say, I'm not big on just publishing papers. I do have quite a lot of them. But the reason for that is because if you don't publish it, no one will know that you did it. All right. But you also have to get out there and talk about it. So think about how to make your research impactful and engage the public, the press, and policymakers can be one way. So my last slide here. This is one of my, sure it's in the new airport after they remodeled, but it was up for a long time in Johannesburg in South Africa. It's an African saying, if you want to go fast, go alone. If you want to go far, go together. And that's a big part of the One Health approach. Thank you. So I went a little over, but we'll attribute that to technical challenges, huh?
Thank you very much. We are going to continue with the Q&A session. It's going to be an interactive one. Anybody joining here, you can just raise up your hand and then the microphone will go to you. And also for those who are joining us through Zoom or Facebook Live, or you can actually drop your questions in the chat box and then we'll take note of it and then we'll relay it to Professor Pat. Okay, so anybody would want to start? Dr. Puan? Yeah.
Okay, I'm just curious. For the parasite, they are attacking the sea otters. I know that the killer whales are also eating the sea otters. So do the whales also get it?
So you said that who's eating them? The Eskimos?
No, I'm saying that killer whales.
Oh, killer whales.
I know they feed on the sea otters.
Yes.
Yeah.
Okay. Yeah. So you brought up, there we go. We got a really ecologist wildlife person here. Yes. So one of the new challenges, and it hasn't become quite as big a problem down in California, but it might, because we're getting increasing numbers of killer whales. All right. Down there is killer whales taking out sea otters. That is a very good question, because I don't know that there's no reason why they wouldn't be infected, all right, with Toxoplasma or Sarcocystis neurona, either one, because we've seen it in a wide range, both parasites, in a wide range of marine mammals, all right. But to my knowledge, we haven't looked at killer whales so much, but that there's no reason why not. So I will raise that to my colleagues as soon as I get back. Because who's been doing the killer whales? All right. because I think they are going to start getting some off the coast of California, but certainly it's been better recognized up north Alaska and that part that the killer whales are a major contributor to the decrease in sea otters. Down where we are, sharks, okay, which, you know, are not mammals, they're fish, right? You know more than me, but I know they're not mammals. And they kill otters, but they don't seem to want to eat them. All right. So sharks, and we have these great white sharks, treat sea otters kind of like they do humans. All right. They may get interested and they go for a bite, which for us and otters probably means the end of our life, right? Or the end of an appendage, but they don't seem to eat them. All right. So and they're not marine mammals. They're not warm-blooded in that way. So good question. We'll look into that. Keep asking those questions. Yes.
Morning.
Morning.
My name is Kenny. I'm from University of Nottingham.
Lovely.
It's an interesting talk. So I would like to actually have two questions. So the first question is about the oocytes from the oocytes from Toxoplasma. So your story is telling that.
Wait, it went off for a second.
So the parasite actually survived from fresh water until the salt water in the sea, along the journey, long way, and that's a long time.
It's okay. If you say it, I can repeat it for you.
Okay, so to my understanding that I don't think they could. That, I mean, the lifetime of the parasite is that long.
Okay, so can I answer that one first? So the point that's being made is that if you're talking about oocysts shed in the feces of cats, all right, they getting into fresh water and coming to the ocean to infect marine mammals, that seems like a very long time. All right, so a lot of travel. All right, well, first, just want to make clear there are also people feeding feral cats right on beaches. All right, in California and other places. Hawaii, there are big cat sanctuaries, and some of those are not that far from beaches, and it's Toxoplasma is becoming a major cause of death of monk seals, which are endangered. All right. So sometimes it's right there, but also the point I didn't get to mention that partly answers your question is that these oocysts are so tough. You would not believe them. You can actually take oocysts of Toxoplasma, and people have done this, and store them in formalin. All right. So 10% formalin for years. Take them out, wash them off, and they will sporulate and be infective. All right. So yeah, that's how tough. They're tough. I think they're at least as tough, if not tougher, than ascarid eggs. They're very, very tough. All right. And and they take very cold, a lot of studies, a lot in the literature, they can take really quite cold water and they can survive for years in the soil. We know that. Because studies were done in Costa Rica and in various places. So yes, it's conceivable that oocysts can travel quite a long ways, even outside of California, because everything goes one way or another into rivers and oceans in the United States. I mean, from the top mountains, everything eventually goes there that isn't into the ground or drink or up. You know, that's the kind of flow. So good question. Gave me a chance to make that point. These are very, very tough. All right. You had a second question too.
In Malaysia, we do have other kind of outbreak reported, like on the island resort. The so-called found yet, it was possible to be connected. So we wonder
Oh, good. So let me repeat that. But we should get a working microscope so the name of so the question is about Sarcocystis. So Sarcocystis neurona, all right, can be cultured continuously in vitro. All right. We've done it. A lot of other people have done it. All right. But there are other Sarcocystis parasites and so the question is, could they be cultured, all right, in vitro? Because there's a specific one that you're interested in here in Malaysia. And what's the species name? Sarcocystis nobilis. We need to get a microscope. He's better than me. So I would give that a try. Okay. So we learned to grow parasites, Toxoplasma. Nobody had ever isolated Neospora, but we just got good growing Toxoplasma, and then we applied, and I used to grow Theileria. Okay. So if you have people doing tissue culture, giving that a try is, I think, very valuable. The thing about Sarcocystis, most of the Sarcocystis that are in their intermediate host, they're in cyst stages. So you have to have a way to break open the cyst. I mean, or give them a chance to come out and grow, because it's that, and sort of reactivate. It's that stage. All right. And whereas with Sarcocystis neurona, we oftentimes they're free in there. They haven't insisted always in the horse. So, but it's doable. I mean, they found ways with Toxoplasma to culture from cysts. So I think it would be, if it's important, give it a shot. Okay. And sometimes, giving it as a graduate student project, but don't make it their only project, because that was I wanted to culture Theileria and red blood cells. And I spent most of my PhD doing that, and it absolutely didn't work. But you should see my thesis. It's about that thick, full of negative results. All right. Fortunately, I did get a few things. I never got the culture going, but I did learn things that I was able to write papers about. So, but yeah, good. I'd love to hear it. And we haven't done those exactly, but I would encourage you to try.
Yeah. Thank you.
One question for the folks out there in the global verse.
Oh, okay.
Why don't you ask it?
Yeah. So I'll just read the question. It's from Dr. Azlina. So as a young lecturer, what should be our focus and how to become successful and remain relevant in academic settings?
Okay. Well, that's kind of why I tell my story, because I had exactly that same question. All right. When I arrived, I didn't know, what I could or should work on. For me, I think it's a very personal situation, but I can say for me, what helped was being a veterinarian, but I think the same true is for non-veterinary scientists. I like to solve problems. Okay. I like to work on things or I feel highly motivated to work on things that are creating a problem that people want to know how to fix it. All right. Now, I may not be able to fix it, and I may be coming in just trying to figure out what the parasite is in the beginning. But once you have the parasite, then there's a lot of other things that you can do. Now, the trick there is it's very hard to get funding for something that people don't know about yet, right? So that's why I'm a big advocate of collaboration, is that I wouldn't just sit in your office or sit in your lab thinking, oh my gosh, what should I be working on? Be talking with people, all right? Be thinking more broadly. Make lists of, here are some possibilities. And then what excites me, all right? What's going to keep me going? Because any of us who've ever done research know that there's a lot of frustration, right? There's a lot of things that don't work, all right? But if we believe in what we're trying to do, or maybe we're not quite sure if we should believe it, but we know it's exciting to think about. You've got some good ideas, or your team has good ideas, or both. Then that's helped me a lot. And get mentors. All right. People that, this is why I said to Dr. Farina that of all the things, I'm not as interested in giving lectures to students. You all can do that. All right. But if I can share my experiences and give any kind of guidance or mentorship, support, connections with other people that can help to advance your career. All right, then, and it's not just women. Okay. But they oftentimes need extra help, but I have 2 sons. I had 5 brothers. I'm a big supporter of everybody. Okay. Of moving forward. So, but I just bottom line, I see, having something that you feel passionate about. That's why I showed the example, as silly as it might appear, of my affection for East Coast fever. All right. And I'm still excited about it. I'm on an advisory committee for a project in Kenya. It's quite amazing because I still get excited about it. But I worked on a lot of other things I'm excited about, but also especially if you're a problem solving person, what could possibly make a difference? But having said that, just one little qualifier, a lot of people, you may be working on fundamental things that you can't see now how they're going to make a difference. And that there's lots of examples in the history of science where those fundamental studies that someone was doing just because they were interested in them turn out to be the big breakthrough that allow us to develop the vaccine or develop things that we never imagined that have had a positive impact on the health of people and animals and the environment. There are a lot of examples. Okay. And I got a real recommendation, a really good podcast when I was in the UK that sort of has a lot of top scientists telling that story. You wouldn't believe how many times people started on something because they had coffee with somebody and they thought, hey, what about this? Or they themselves got excited about it and they worked on it. And down the road, it turns out that maybe with the help of somebody else ends up maybe on their own, ends up being the big breakthrough that we are all taking the advantage of now. So I hope that helps.
Hi, Prof. I've got a bigger question. Okay, just let me stand up. Can you show us the slides where you have all the next after you, the researchers after you? So this is at the level where we want to encourage the next layer of researchers, postgraduate students, or people to be interested in this field of research.
So you want me to pull up that slide?
Yeah,
I sort of sprinkled them all the way through there, but I did have that one with the 3 ladies.
Yeah, the 3 ladies. So that means you have like a layer of researchers or colleagues that continue on the field of parasitology for you. And this is where the legacy and continuity of the field are being carried over time, right?
Yes.
So the challenge that we're facing now, it's to encourage people to go into research. And for veterinarians, and this is a problem when you are preparing for the next layer and there's no next layer. People are just not interested to go into research. And that's one thing. Second, the reality of the situation where, of course, this is pertaining to veterinarians, yeah? The moment you graduated, it's very lucrative to go direct into practice. Of course, because of the business itself and the environment in the academia world in which you've got to sacrifice first.
Yes.
Before you're able to reach there, and it's a risk. You see, you may finish or you may not finish, or you may make an impact or you may not make an impact. So these are the things of how would you I think encourage alone will not be enough, no? Because this is people's choices in life to pursue academic world, to go into research. So may I hear your opinion on this? Thank you.
Absolutely. So on that point, especially let's take veterinarians. All 3 of these young women who now have very successful careers, they're all veterinarians. That's what the DVM, Doctor of Veterinary Medicine, stands for. Okay. And they at least these 3 women, they all did a year of at least maybe 2 years in practice. All right. Because the clinicians, when they got to their clinical training in the last 2 years, the clinicians are always saying, oh, you need to practice. No matter what you want to do, you need to practice. You need to practice. You need to practice. They told me that too. All right. You need to practice. I intended to practice. Okay. In my case, I didn't, because I got a scholarship that took me on a very different path. And I never looked back after that. There were a few times when it was kind of rough, and I thought, I can always be a vet. All right. That was kind of consoling. But these 3 women, when they came to me, they'd had a look at practice, and they knew that they were not going to be happy forever. All right. With that, they knew that it wasn't intellectually for them. All right. Whatever their reasons, you'd have to talk to them exactly. But mostly they may not have thought they were going to be academics, but they knew they wanted to come back and explore research. All right. And once they got in there and they found things that they liked, they knew, yeah, this is the way I want to go. And it's not been without some challenges. In all cases, these 3 women that I just happened to show here, they all had husbands, all right, that had careers, and they made decisions to that were important for them as a couple. All right. So that in some cases, slowed up their career or changed where they went or whatever, but they had supportive husbands, and they ultimately have all gotten into really good positions. I also want to mention that although they were, actually Liz worked with us, but she was Jonna Mazette, who you may know from One Health. She was her PhD student, but all of these women would going to call themselves One Health practitioners, because they don't just work on parasites. They all work on viruses, bacteria, other infectious agents, and beyond, environmental problems. They're working on all kinds of things, because that's the exciting thing about this generation, your generation, is a lot of people say, well, I don't want to just work on the same thing forever. I saw my major professor did that. Yeah, well, maybe that was, you saw I didn't do that. I would never have worked on one thing forever. All right. So, but sometimes that what used to be the thinking. All right. You go in academia, you get on a track, you stay on that track, but not this new generation. All right. So don't be afraid of considering academia because you think you'll get locked in to doing the same thing forever. You can be, I think, better than in any other field. All right. Be your own boss. All right. So where yes, you may have some people above you, but if you're doing good work, I've found, I've never had a problem with that, because this has created the right environment. I tell a story because I never imagined I would have had the career that I did ever. All right. But as it went on, I did it because I love asking questions. I love discovering things. I love working with other people. So does that answer your question? Does that help? Okay.
For the world of the research, when you are meant for it, when you found it and it's for you. So at my level, if let's say I were to inspire other people to come and join the research world, and if it's meant for them, they'll come and just stay because they found it. Yep. Right?
Yes. And also the one thing I try to do with my graduate students is that, and I got better over time to do that. My first graduate student, that came from Japan, we just had a problem. We needed to get going on it. He liked it. We did it. All right. But he had input. He had a lot of his own ideas, but sometimes coming up with your own ideas is challenging. With all of these women and others, Ann Kimtrup, I showed you her picture as well. And all of them, I try to say, okay, this is what we're doing. This is what I know about, but what do you want to do? All right. What are your ideas? But then help them to take responsibility to actually come in and help me write grants. Okay. Help me find the funding. All right. Because People do their own ideas best. All right. So it's aligned with mine because that's how I can best help them. But sometimes it's not. I have one that came also, gone out to practice, came back right away. And she worked on bacteria in elephant seals. Okay. We didn't have a bacteriologist. So I was her major professor. Now she's a scientist at the CDC. All right. And she never thought she'd end up there, but she loves it. So she gets to do research and travel around the world helping on public health problems. And so when you think, I don't want to be an academic because it looks like this, don't look at us and say it looks like that. You have a chance to create the world that you want. It may not be easy. Mine wasn't easy to get people, but if it's in your heart, if you're listening, as you said, if you're listening to your head, what interests you, you're listening to your heart, what feels like the right thing for me to be doing. Okay. Then I think that's how you help. That is how you find your path. And good mentors. Yes.
Thank you, Pat, for the very interesting and impactful life journey. And congratulations. I think you have made your life happy. Yeah, the thing is that about the One Health, see, your sharing has really triggered me to review on what understanding that I have really on One Health. Because all this while we have been, I'm from the ruminant section, and I teach these farmers to do good practice so that, one day they can get MyGap. Yeah, MyGap is just a scheme whereby the products of the animal, the food animal that they produce, would be of quality, and they are also safe and wholesome for human consumption. So it's not only the vets that does this to get MyGap, it's also the agriculturists and also the fisheries people. So that's the Malaysian way of trying to make sure that the food that we produce are all safe and wholesome. So we, in order to achieve this, we make sure that they look at the environment to make sure that the slurry doesn't get out, and they should go into a pond, and only when the water is safe, then only you let it out into the rivers and so on. Then the environment, the animal must be healthy, must be happy in actual fact, just like what we want to be having, aircon and everything so that we feel happy. So the animals need that too. The animals, we tell them to give balanced food so that all the products that they produce will be of quality, wholesome, and safe. And not only that, when they put in the health input, we give them a vaccination. We make sure that when there's antibiotics given, there is what do you call withdrawal period being looked at, so that there's no residue of antibiotics, so that there will not be problem of AMR. So all this in our world is One Health.
Yes.
So I used to be introduced to, I was introduced to One Health with zoonosis.
Yep.
Of course, is one angle. But now I realize that whatever we are doing, the agriculturists are doing, and also the fisheries are doing, is also contributing towards One Health.
Absolutely.
And that is really great.
Oh,
Thank you very much.
Yeah, I think you've said it beautifully. Beautifully. Yeah, no, some of our closest collaborators are in the College of Agriculture and Environmental Science, and also beyond that. I mean, I'm on an advisory committee, and I've tried not to do too many things, and I was retired. I mean, the East Coast Fever, coming here, and then I'm on an advisory committee for global agricultural sustainability for the University of Edinburgh, and they are definitely thinking like you. How do we get this into the conversation? And it's not all about zoonotic disease. That got a lot of attention, but no, people in agriculture leading the way in One Health, because you get it. You are like the vets, you get it. The medical people struggle a little bit because but we have some really great advocates in global health, public health as well. So good. Thank you for that. You should give the next talk.
Thank you for the pictures of CITBN and Duncan Brown.
Oh yeah, yeah. So I have met a number of other alums of the University of Edinburgh, especially the CTVM. So yes, so the building looks the same now, but it's kind of, I don't know how long they're going to let it stay. They built the Roslin Institute, which is just spectacular. It's right there too. So yeah, it's an amazing universe and university. Any other questions? Yes.
Thank you, Pat, for the amazing talk. Well, I'm curious because I've actually got 6 cats at home. And, you know, when we remove the poo, is it better to wear like a mask then because of this toxoplasmosis?
So let me ask the first question. So you have 6 cats. Are they indoors all the time, or do they go? They're outdoor cats. Okay, yeah. So thank you for giving me this opportunity to mention that very important point, is that everybody talks about cat litter. All right, the stuff that you put in the cat box as being the source. Even we've been having this battle for decades, because especially reporters and things like that, everybody wants to talk about cat litter. There's cat litter that you buy in a bag and you put in a box and your cats poop in. That's not a problem. It's the cat poop that is the problem. All right. So if your cats are indoors all the time, and I don't know what percentage of people in Malaysia keep their cats indoors all the time, not too many in California. All right. They somehow think it's really important to let their cats out, including, I had 2 outdoor cats. Okay. So I'm not pointing fingers. All right. I am guilty. But if you can keep your cat indoors, so it's just using the cat litter box, and you're feeding the cat. That's the other really important thing, because where is the cat going to get toxoplasma? Not from canned or bagged cat food. If it's properly made, they're going to get it from eating birds and small mammals. And so that's why indoor cats fed by you with a cat box is not a big problem. I would say, no, you don't need to wear a mask for that if they never go outside. All right. Cats outside, or those that go inside or outside, we know even indoor, outdoor cats, even if you feed them, they like killing birds. Okay. They like killing small rodents. And so that is how they can get infected. So if that's the case, then you have to sort of view it as as potentially contaminated. Now, but here's really important, getting into the fine details here. Cats that are infected, there's a lot of great data to show this. When they get infected, they generally, and this is a big, really like almost all the time, shed one time. All right, the first time they get infected. But when they shed, they can shed millions, up to, we've had 1 billion, all right, of these oocysts. But it's the first time they get infected. After that, they don't shed oocysts. They just are a regular intermediate host. So what does that mean? That means that the safest cats are indoor cats all the time, or if you had to choose between getting an older cat or a kitten that's going to be in and out, go with the older cat, because they probably have already been infected. All right, if they've been indoor, outdoor. So they get infected once, they shed large amount, and then they are immune. They're at far greater risk if they're eating birds and small mammals that are the source of infection. All right. So who's at risk of getting disease with toxoplasma? What? Humans, women. All right, pregnant women. And so I tell our veterinary students, look, if there's, which are now 88% or more women, if you, as a cat owner or a veterinarian, if there's any chance you might be getting pregnant, and sometimes people get pregnant before they have, planned. Okay. But if there's any chance, if you're reproductively, a reproductively capable woman, and you might get, then be particularly aware of cat feces. All right. And it's a good time to train your partner to clean the cat box. Okay. That's right. They all love that. But also remember that it's the contamination of the outdoor environment that you need to be aware of. All right. So like plants, like vegetables that are grown in soil and water, and any of those things that could get fecal contamination in the environment is a much more likely source of infection. All right. So, and what we should all try not to get toxoplasma, because at any point people can get immunosuppressed, and then that can also be a problem. Does that answer your question? Okay. Yes.
Hey, Fat. I'm Atika from the vet faculty. So I'm doing my master's right now, but I believe that my results might came out negative. So from your experience, you actually get a lot of negative results in your PhD. So how do you actually justify all your negative results?
Yeah. So negative results, there's nothing wrong with negative results. We don't like it. Doesn't feel good. But remember, science is based on a hypothesis. You make observations, you form a hypothesis, right? Which is this like a question, right? That must be testable, right? And if that test shows that hypothesis was wrong, all right, then if you designed the question right and tested it, it should lead you to the next step of what you need to do or want to do to try to figure out why it's not what you thought it was. All right. Now, if your project is to grow parasites in a culture, all right, and they're not growing, you keep asking that question, they're still not growing in the culture. There comes a point that you just, maybe they just don't grow in culture, all right? But so how do you so I would say negative results are justified, all right, especially if you've thought them through. Now, the problem is publishing negative results, all right? Nobody wants, very few places will publish negative results. All right. We did have that dilemma once when we were working on the vaccine for Neospora, and it showed that despite what the company was trying to say that was funding us, it was not efficacious. And we thought that needed to get out and be published. All right. And that was the thing we were facing. Fortunately, I was sharing that problem with a colleague, and right behind me was the editor of the International Journal of Parasitology. And he said, "Oh, we'll publish that." If it's negative, it's all right. It's important. We'll do it. All right. So that's my one, very positive experience. So of publishing negative results. So you're doing a master's. Yeah, we could sit down and look at them, how, because there's a requirement to actually publish a paper, right, for the master's. All right. So sometimes in the course of doing something that's not going the way you want, you can still learn things. So be open-minded. Don't be stuck on, this is only if it works my way. All right. There may be other ways to look at the question, and maybe you already have some of the data that will guide you, okay, to wrapping that up in a way that you can publish. In my case, I was trying to grow the Theileria stages in red blood cells, all right, because they could do it with Babesia bovis into continuous cultures. That did not work, but I did get them to divide, all right, and with Theileria parva, they divided and they stayed in the red blood cells into those Maltese cross forms. And at that time, all the literature said that Theileria parva did not divide in the red blood cells, all right, like 50 years of literature. And I was looking at it, and we did electron microscopy. I learned how to do that. And they were dividing. They were dividing by schizons by schizogony into those merozoites, like Plasmodium does, okay, in red blood cells sometimes. Anyway, so my point was that I thought, wait, but this is against all the literature. That's another thing that's hard to publish when you're going against the literature, but so important that you persevere. So we studied it. We could show how they divide. And then when I looked back further and further, there's an important lesson in this little bit here too. Further, but at the insistence of Duncan Brown, go back and look at the old literature, which we don't always do. I saw that Robert Koch, who first described, Theileria, and then some other people, they drew the same forms, and they said they were dividing forms. It just is somewhere along the line, somebody started to saying, though, they didn't divide. Those weren't dividing forms. Okay. So that's what was one of the papers. And then I did it with Theileria annulata. And then I had a third paper about Babesia bovis, because I had that as a control and I could do an experiment there. So I pulled that off. I got my 3 publications, and in the British system, we had to write that thesis, so negative results counted. But anyway, does that make sense? So there's a lot of different ways. So don't be discouraged, okay? Sometimes the world is not the way we think it should be, and sometimes things happen for reasons that we haven't thought of. So that's the fun of being a scientist.
And so, hi, Prof. It's not a question, but just an acknowledgement. I would just like to say thank you so much for listening to my project last week. It really gave me a lot of courage, because I really thought it didn't have any value, because I only started to do the research just based on my interest. So yeah, thank you so much for attending and giving me all the support that you have.
Oh,
That's
you're
all.
very welcome. Thank you.
Oh, and I'm happy that you managed to see the otters last night.
Yes, because your talk was the otters, right? Yes. Oh, she gave a great talk. I was early, and then she answered the question so well, because she said, future studies, we should look at what otters can give to humans and other animals. And I said, yes, but what can humans and other animals give to otters? And she knew the whole Toxoplasma sea otter story. It was amazing. I was thrilled. So yes, they are amazing. And one of the highlights of my time here was to be able to have 2 days that I was able to listen to a lot of the final year research projects by the veterinary students. And I really enjoyed that, because it is just a 5-week period that you have to do your research. But I think that was a great introduction to not just how to confront research, but how to make presentations, because the students did amazingly well. So thank you for letting me listen. Yes.
So when you talk about One Health, it's going to be involving a whole lot of different entities, different parties. So what would be your biggest tip or advice in order to keep working with collaborators and potentially competitors in harmony? Because conflict may arise. So what would be your best advice?
Whoa, there we go. Okay. Conflict management, important life skill. All right. So it's not just scientists, it's everywhere. It's in our personal lives, it's in our professional. I don't think anybody can escape that. All right. And you're so right. How do we get good at that? There is no secret formula. All right. I think for that, I've done, a dynamic dialogue classes and courses for a couple of years and other things. So important. So special tips. One, recognize that it's important. All right. And don't give up. All right. Don't just go silent. All right. You don't have to go silent. You don't have to be combative. You don't have to do anything. It starts, I think, with listening. All right. Listen to what the other people, especially if there seems to, under any circumstance, listen and be sure that you're hearing. All right, what the other person's trying to say. Remember, we all have biases, and we make decisions sometimes that prevent us from carefully hearing what the other person has to say. Ask questions, all right? Really try to understand their perspective, right? You might have to do that. You may not be able to do that in a meeting. You might have to do that in a sideline, all right? But if you can understand where they're coming, and if they felt heard by you, that makes a big difference, right? I mean, for all the professionally and personally, people, feel like they're being heard. And then don't give up. All right. I'm giving a simplified version here, but don't give up, because another thing that I think is also, try not to take it personally. Because a lot of times when there's a conflict, all right, we can't help it. We think, this is between us. This is a conflict. And sometimes it's not. I like to remember everybody, when you see other people, everybody is dealing with other things in your life, that in their lives, that you know nothing about. All right. And if you can have that compassion, all right, that doesn't mean you have to cave in, you have to give up or anything like that, but trying to understand, listen to, feel like, allow people to feel like they're being heard, understand their perspective, you know. And then there are there's, again, it's a getting mentored can be very helpful, because look at the people that you know, and oftentimes those are older and wiser people, but not always. Sometimes they're not older, but look for the wiser ones that can maybe help you to talk things through or figure them out. But you need to do that oftentimes in a way that's not going to make the problem worse. There got to be people that you can trust with confidence, that you can share the situation with. Does that help? There's a lot of other tips, but I think those are sort of the fundamental ones. And I'm what I shared before. When it comes to collaboration, I know you're always going to hear some people say they had a terrible experience, and I'm telling you my experience. I have had some bad experiences, but what I learned from that is if at all possible, and as you get older and more senior, it becomes very possible. I work with people that they may or may not have all the skills I want, but that I trust, and that trust me, right? And this communication is a big part of the trust. Okay, hope that's helpful.
So I'm just going to give one last chance for those here, if there is any burning questions. Okay. Otherwise, Prof. Patricia will still be here around. She'll fly back to the US on Friday. Okay, so you're always welcome to drop by at the faculty. So, thank you very much, Prof. Patricia. But I would like you to remain on stage. Words can never be enough to thank and to express our appreciation and gratitude. So I would like to invite Dr. Farina Mustafa Kamal to come forward and to hand over our tiny gift token of appreciation for all that you've done to us in the past few days, and also for today. Okay. All right. We would also like to invite Associate Professor Dr. Stikarani to come forward as well.
Oh, yes.
Yeah, our former cute head of department. We get photographs. A big round of applause. Thank you. And I think we'll have a big photo, one group photo. So I would like invite everyone to come to the center of the stage. So you can just kind of like sit down. Yeah. And you can have a photographer. Yeah. So it, the photographer can take it from here. Jump. Come, Ika. Okay, we've got everybody. Tak nampak ke? Oh, tak nampak. Okay. So you can squeeze to the, yeah, to make sure that you'll be visible. Okay. Freestyle. Peace, all right. Yay. Okay. Thank you very much. So I'm pretty sure we have learned a lot during this session today. What I know is that I think I'm going to print a t-shirt with 3 words on it: tiny, tough, and mysterious, resembling the toxoplasma that was shared by a professor just now. So I really hope so. Okay, so to all of the attendees here at the auditorium, you can grab a pack of food. I think it's kueh as you walk outside the hall. And thank you very much again for joining us, and those online as well. So you can actually catch up the session on MyHoon's YouTube channel and also Facebook page. Okay, thank you very much. Thank you again. Assalamualaikum.